Thank you, Kendra, and thank you, Ilse, for inviting me. And wow, 25 years of experience. I dare not ask how much you have. I am on the other side of the spectrum, a very green assistant professor at Cornell. And I should also thank my colleague, Dr. Martin Weidman, who was originally slated to give this talk, but couldn't make it and tapped me on the shoulder to give this. And so some of what I'll be talking about today, and I should probably bring up my presentation, is going to be, so what I'll be talking about is, some of it will be covering Martin Weidman's work in the Department of Food Science, and also some of our other colleagues, Sam Nugent, their work in bacteriophage, and some of the work that I've done. And really, the goal of this is to give you an overview of bacteriophage, how we use them to control pathogens in foods, some of the challenges in that application, and then some of the opportunities. So to begin with, bacteriophage are viruses that infect bacteria. And if the pointer here doesn't quite work, but they're composed of a capsid, a tail, which is that long cylindrical part below the head, and then tail fibers that allow them to attach to the bacterial cell wall. It injects its DNA into the cell, hijacking the metabolic machinery into producing more phage. It also releases some holins and lysins that disrupt the cell wall and allow the release of the progeny to go on and infect more bacteria in the area. Now, bacteriophage are thought to be one of the most numerous organisms on the planet, and so they've been around for a long time, but to get a little bit about history, which is always fun, the human history with phage began just before the turn of the 1900s out on the Ganges River in India, where there was a scientist, Ernest Hankin, who was dealing with a cholera outbreak. And he noticed that by taking some filtered water from the river and then exposing it to the cholera, that he observed some antibacterial activity. And he said, well, maybe we should be drinking river water to combat cholera. And that didn't probably quite work back then because they didn't have great filtration systems. And that kind of fell apart. But about 20 years later, these two scientists, we've got Dr. Frederick Twort here on the left. He was doing research in the United Kingdom, and at the time, he was doing work on smallpox vaccines, and one of the challenges with smallpox vaccines back then was that it was made using the skin of calves, and it was very susceptible to contamination by Staphylococcus. And so he was looking at these Staphylococcus colonies under the microscope. There were these big colonies, and he noticed that some of them had these kind of white glassy specks in them. And so he tried to culture these specks, and he couldn't get them to grow. But then he found that if he took those specks and then touched them to another colony, that these little glassy spots would actually develop in those other colonies. He did some more work finding that he could filter, that these whatever these agents were could pass through a filter, and he could take that liquid and then also get these glassy spots to develop on other colonies. And he thought that this was some sort of bacteriolytic agent, maybe an enzyme. He didn't quite know what it was. And then unfortunately for him, World War I started, and he lost funding to do this research, and he never went back to pursue it. Now, he did publish some of that work, and around the same time, the other gentleman on this slide is Dr. Félix de Herelle. He's a French-Canadian scientist who was doing work in Paris. They had an issue with some of the troops stationed there. They had an outbreak of dysentery. And so he was looking at cell-free lysates of Shigella and found that if he applied some of that filtered cell-free lysate back onto plates with Shigella, that he started to see these clearing patches, which he ended up calling plaques, and which is now why we call this the plaque assay method. And he really actually saw beyond this. He wasn't just seeing spots, he really understood the potential applications that this could mean for treating diseases in humans, and he became one of the strongest advocates for phage therapy at that time, and he traveled around the world, Brazil, he set up a research institute in Georgia, looking at identifying bacteriophage to combat disease. Unfortunately for Felix, there are a couple things that came along. Number one, around the same, a little bit later, there came the development of penicillin. And while people were trying to propagate phage, they didn't know a lot about the biology and the requirements to do successful propagation, and so they had a lot of challenges, and this was much easier and had a broader action. Also, I mentioned that he started that research institute in Georgia, and after World War II, that kind of ended up on the wrong side of the curtain, and so there are a lot of different factors that kind of led to the kind of disappearance of phage as an antimicrobial agent in Western medicine. Phage did come back into popularity in the 60s and 70s from the molecular microbiology side of things, where they were studying viral host interactions, and it led to things like the T7 constructs that we now use in molecular microbiology, lambda phage biology, and things like that. But as far as an antimicrobial agent, it hasn't really been that popular until relatively recently. I don't know if people get this in their Twitter feed, but this issue with increased antimicrobial resistance to now common antibiotics has really driven a search for alternatives to our common antibiotics. And this came out in the Huffington Post earlier this year. Now people are beginning to look back at bacteriophages as a possible solution, right? So hey, look for sewage, it could save your life, which is kind of fun. And so why are we looking at phage again? So again, back to this little drawing. The reason we're interested in phage is, unlike antibiotics, they can be fairly specific. So they can be targeted against a species of bacteria or a strain of bacteria. They're rapid, right? This infection, the lysis cycle, happens in as little as 20 minutes. All right, and then lastly, they're self-dosing, right? So if their target host is present, they're gonna make more phage that can go on then and kill any other potential hosts that are present, as opposed to antibiotics, where you've got to take that nasty little pill, every 8 hours to make sure that you can actually eliminate the infection. So this is why people are back looking at bacteriophage. And it's also trickled down now into food, where we're looking at applications to use this to control pathogens in foods. And there's a few commercially available products now. There's Interlytics, which has a series of products. They have List Shield, which targets Listeria. They have EcoShield and Salmo Fresh, which target E. coli and Salmonella as well. There's Listex, that's at a company in Europe. And these are all geared to the food, human pathogens. There's actually also a company here in the US called Agrifage, which is developing phage products to target pathogens for plants. And so this is really broadening out there. And I'll go into some of the examples of this use in foods. So here's an example. This is a paper published by the group at Interlytics a couple years ago in the Journal of Food Microbiology, where they looked at contaminating different foods with Listeria, and then applying their List Shield cocktail. And so some of the things to note here is, so they looked at applying this in Listeria, and you see that after 48 hours, they noticed a reduction in Listeria counts, ditto for the smoked salmon. And they also mentioned a dinner entree. They didn't quite talk about its composition, but in all cases, they showed that within 48 hours, the application of this phage cocktail that they have, which is composed of 6 different phage strains, reduced the levels of Listeria. Now, one of the things you note in the way that they report this is that they talk about different inoculum levels of the phage. And this is pretty important when you're thinking about applying bacteriophage to your foods, right? Phage levels matter because you've got to find the bacteria. And so if you think about a mill of water, and if we were to blow it up to, say, Loch Ness, right? And we're looking for the Loch Ness monster, which is really hard to find, our Listeria, that's somewhere out there, you're really trying to find that Loch Ness monster with a watermelon, right? So you're gonna need a lot of watermelons to find that in an effective amount of time. And now when we talk about water systems, there's things like mixing, and phage can't swim, direct themselves, but there's Brownian motion. So there's some potential for the phage to find the cell. But not all our foods are liquid, right? So you imagine if we were actually applying it to a food that was hilly, like those mountains on the side there, that phage can't travel anywhere. So the amount of phage that you're applying is duly important for making sure you actually reach the bacteria and kill them. And so there was a study out of this other group where they actually started to look into this. They looked at 3 different fruits, melon, pear, and apple, and they looked at sliced versions of this fruit, and then they looked at juiced versions of this fruit. And they had a control here. They inoculated it with, here they were using Listex, so the P100, and they monitored the Listeria levels over time in both the sliced and the juice, and then also the phage levels here with these little triangles. And what you can see is that there's definitely a difference depending on the food matrix, right? So for the melon, while there was some inhibition of outgrowth of Listeria, it was definitely muted compared to the juice version, where we see a very rapid die-off of the Listeria for the melon. And if we look back here at the pear, there's a similar story there, again, where you see a difference between the sliced version and the fruit. And when we go down to the apple, you see, at least for this study, they didn't really see any difference in inhibition with the phage present or not present compared to the control. And they also saw a pretty big die-off or disappearance of phage in their solution, which goes to the matter, they thought this was probably due to pH, but it speaks to the fact that the food type that you're trying to treat with phage matters, and you need to take this into consideration and do some validation studies to make sure that the phage are actually doing what you're hoping they're doing in your food. Here's another example from a different group where they're also looking to see if they could use bacteriophage in synergy with other antimicrobial compounds. And here they were looking at potassium lactate and in the application in deli meats. This is some smoked turkey. And what you see is this top line here is their control with no phage or PL added. And you see the Listeria growing. The A is at 4 degrees and the B is at 10 degrees Celsius. And when they add the potassium lactate, they don't really see much difference in growth, but when they add the Listex, there is a die-off, but then there's eventual little regrowth, right? After about 7 days, Listeria starts to pop back up, and while the combination of the two seem to work better, you begin to see a little more increase, and the story's kind of a little the same in the 10 degrees. And the thing here is to remember that these are hurdle controls, and they're not here to wipe out the bacteria that are completely present in the food, and so shelf life matters. So if you're thinking that bacteriophage are going to provide you a completely safe product over the entire shelf life, you've got to think twice. So you've got to understand what's happening in your food. And that story also, then here they looked at roast beef, and you can see that the story changes a little bit, but it's overall the same. They mentioned a little bit there about some differences in temperature. In the Weidman lab, they did some work looking at some phages that infect Listeria and looking at the effect of temperature on the ability for plaquing and adhesion. And so here, I know this is kind of small, but they looked at 3 different phages that were specific for Listeria monocytogenes and 2 different strains of Listeria that were present in the lab. And they looked at them at 30 degrees C and 37 degrees C. And you can see that there's a clear difference between both on temperature, wrong way, on the plaquing ability based on temperature and between strains, right? So depending on the strain that's present, there can be differences in the efficacy of your phage treatment. And they also looked at the impact on adhesion. And here again, you can see that there's differences. This is one of the phages, and there's this second phage against different Listeria strains. And there's varying difference based on the temperature, whether it's 30 degrees C or 37 degrees C, and how well they're able to absorb to the cell. So the temperature of your food, your process, is also going to impact the efficacy of your phage treatment. So you need to take this into consideration. One of the things is when we talk about using phage to treat Listeria, say, in the environment, right, we have big issues with environmental contamination from Listeria monocytogenes. The Weidman lab had done a study from 1998 to 2009 where they were characterizing Listeria strains isolated from the environment and looking for persistent strains in this smoked seafood plant over time. They went back to this study and pulled out those isolates and looked at the way these isolates were inhibited by one of those commercially available phage cocktails. And what you can see is that depending on the strain, so here's 3 strains that were persistent strains in this seafood plant, and this is one of the lab strains, that some of the persistent strains were, this is the control, and then they increased levels of the phage that they added, MOI of 1, 10, and 100. And some of these were susceptible, but several of these persistent strains were very resistant to this phage cocktail. And so, you know, we buy these products or attempt to use phage, but you really have to understand what's there in the environment to ensure that it's effective. Right, so persistent strains can matter. So some challenges, right, that kind of gets to some of the challenges with using phage to control pathogens in foods, right? We know that bacteria are going to develop resistance, right? If they're exposed to lytic phages, that's just the name of the evolutionary game. We know that resistance develops. We also know that in Listeria monocytogenes, it has a functional CRISPR system. It's not clear whether or not that CRISPR system is used to prevent phage infection, but it's there. We also know that physiological conditions can modulate the way the cells work and change how susceptible they are to phage infection. So one of the things that we need to do as we move forward with phage research is really understand what's out there. We don't know a lot about the phage ecology, particularly Listeriophage. So the Weidman group did a study where they isolated phage from the environment, that was specific for Listeria, and they did whole genome sequencing and then characterized these strains. And what they found is that these strains, the strains that they found could be organized into 5 conserved regions based on their gene content and their morphology. And you can see that there were some here, the Myoviridae, and then a couple different clades here in the Syphoviridae. And the interesting part here is that the breadth of activity varied among these phages. And when we talk about the phages that we've been currently doing a lot of research on and are in the commercial market, most of the phages are Myoviridae. So if we have the product there, List Shield product, it's a cocktail of 6 phage, and 5 of those are Myoviridae, and only 1 of them is a Syphoviridae, and the Listex is just the sole phage strain that's a Myoviridae. So there's an opportunity to go out there and better understand what is the biodiversity of Listeriophage, and understand, do they have improved range of infection, and things like that. So now one of the things also to watch out for is we talk about these groups being clustered based on morphology and gene content, but they noticed that as they were analyzing and screening these phages that they had these 2 phages and they looked very morphologically similar, and they were also very genetically similar. They were 99.7% similar genetically, but they showed very different absorption profiles to Listeria. So this one here on the right absorbed very quickly and at high numbers, and then after the lag period went on to do infection, whereas this phage did not. So the question was, what were the differences, and could we look at develop some resistant Listeria and understand what genes were contributing to those differences in adhesion? And so they did this, where they infected L. mono with those 2 phage strains and then selected for resistant mutants, sequenced these with whole genome sequencing, and then went on to identify the differences. And so what did they find? So they found over 80 mutations in total that were kind of located in 5 distinct loci. And if you mapped out specifically the single mutations, they mapped out to about 10 genes in these 5 loci. And as they took a little deeper dive into what some of these single nucleotide mutations were, one of the things that came out, that popped out, was that some of these, and a few of the genes, were in these highly repetitive regions, right? So here, in the promoter region, and another one down here in the actual portion of the gene. And so what they signified is that potentially, right, these mutations led to resistance, right, in phage infection. And because of these repeats, you can imagine that there could be slippage, right, either during, with the promoter or during transcription of the mRNA, which could lead to phase variations that have nothing to actually do with mutations that could then provide, temporary resistance of the cell to the phage, right? And this is kind of still preliminary, but it kind of points in that direction that perhaps phase variation has an impact as well in Listeria, as it's been noted in gram-negative organisms that this can happen quite commonly. It hasn't quite been shown in gram-positives, but there's some reason to believe that resistance to phage can be temporal. They also mapped quite a few of these mutations to 2 genes that were responsible for membrane proteins, and which made them suspect that maybe there was changes just in the cell wall structure that might then inhibit adhesion of these phage to the Listeria. And they did an experiment where, this is kind of complicated, the bottom down here is all the different mutant strains. And then this is the phage absorption rates for LP048 and 125, and these are the actual genes in which the mutation occurred. And so you can see that for a lot of these mutations, they primarily affected the adhesion of this LP125 and not the LP048. Some affected both, and there was one that affected LP0408 more predominantly than LP125. And what they did then is they did complementation tests by then taking the wild type gene and inserting it back into the mutant to see if they could restore then susceptibility, and they were able to do that. And then this kind of pairing study led them to come to a couple of conclusions. The big thing being that they found that the mutants that were resistant to LP125 lacked this N-acetyl glucosamine, which is part of the end of their kind of cell wall, and that seemed to be attributed to the lack of adhesion for LP125, whereas the mutants that were these guys over here that were resistant to both actually had this N-acetylglucosamine, but had changes in the sugar structure of their cell wall, which then also changed the ability of those phage to adhere. So what this kind of gets at is that these closely related phage, they look morphologically similar, they look very genetically similar, but the areas that they attach to on the cell were actually quite different. And so, again, you've got to understand how your phage and your target are interacting. But this also opens up some potential to then maybe do some genetic modification at the way the phage binds to the cell wall, and maybe broaden the ability of these phage to then attach to the cell. Now, so how do we manage, right, all this, the rise of resistance to phage and things like that? And my actual appointment is in dairy fermentation, so I really look at the phage issue now from the other perspective, right? In dairy fermentations, we don't want phage present, right? They kill our bacterial cultures, our yogurt fermentation fails, our cheese fails. And so one of the big things that the culture houses and cheese producers have been working at is how do we get resistant cells? And it's really, a game of cat and mouse and always catching up. And so what you'll see that the culture houses do is that they'll provide a cheesemaker with actually several starter culture strains. So we'll call them here A, B, and C, D, and E. And what they'll tell the cheesemaker is that, if you use culture A for your first, vat or first set of vats, then you should switch to a different culture. And what they've done is they've profiled all these cultures for phage resistance to make sure that they're not all susceptible to the same phage. That way, the idea being that if you're then doing cheese production, is that if you've done cheese production all day long with the same strain, eventually the levels of phage are going to increase to the point where they start killing off all your bacteria and your vats die. Whereas if you start varying the cultures that you're using, and so that they're not susceptible to the same phages, those phage levels never get to the level where they cause a problem. And you can easily imagine transposing this when we're talking about actually using phage to control bacteria in the environment, and instead is developing a rotation program for phage and understanding, right, when resistant listeria then develop in the environment of your food product. Now, one of the challenges here is that in the dairy industry, these culture houses are very large companies, and they have very intensive programs that work closely with their customers. And so every large cheese company is sending back samples from their whey back to these culture houses to look for phage that are present there, so that they can get on top of any changes in susceptibility. And I don't think we have those systems in place yet for managing phage, but it's something that we should be looking for, right? If we want to make sure that we keep this tool effective. Another way to think about phage in my mind is to think of them as kind of nanobots, right? They got DNA, we can program, we can tell them to do things. And so actually one of the projects that we have starting in my lab is we're looking at programming phage to actually produce, and that all went through, antimicrobial compounds, bacteriocins, concurrently with infection, with the idea that is if there's that resistant cell that didn't want to stay there, that's resistant to the phage, that these bacteriocins would then kill that resistant cell. And so you have kind of this double hurdle approach to kind of fight the development of resistance of your target bacteria. Now, we've been talking about using phage as controls for bacterial pathogens in foods, but they can also help us kind of in our control schemes as maybe a mechanism for detection. And so I'd be remiss to not talk about this. So instead of producing, say, a bacteriocin, we could have these phage produce something like a reporter enzyme or reporter protein. So some of the work that I did back in Dr. Sam Eugen's lab at UMass, we looked at getting the phage T7, which is specific for E. coli, to overexpress alkaline phosphatase as a reporter enzyme during infection. And then what would happen is that this alkaline phosphatase would react with BCIP and BT as a substrate, and it would create this insoluble compound that you could then see on a strip. And so what you do is you take your sample, you take phage, add it to your sample that potentially had your E. coli, incubated it for a certain amount of time, and then ran that sample up a lateral flow assay, and then developed that lateral flow assay in a solution with MBT, BCIP, and looked for the development of the line. And so here, this is just a little bit, we did some research with protein MBP that was fluorescently tagged as also a reporter enzyme, and then ALP, and these are the times that we incubated it with the cells, and these are the levels of cells that were originally in the solution. And then for the MBP, we were able to detect 10 to the 3 CFU per mil. With ALP, with a 7-hour incubation and the 2-hour development of the lateral flow assay, we were able to get down to 100 CFU per mil. And then we tried to shorten the time, but then we lost some of our sensitivity. But this is kind of an example of how we could use bacteriophage to detect bacterial pathogens and then control them through testing in the environment. And this is without a concentration step. So now you could work at concentration steps to improve that technology. And this isn't completely new. There's commercial companies out there. Sample 6 has a system that uses a Listeriophage that produces a luciferase that then creates a fluorescent signal that you can then detect for environmental monitoring of Listeria in the environment. And then another final application to talk about is using bacteriophage to help us separate bacteria from a sample, right? Again, in our detection schemes. And so I think many of us are familiar with immunomagnetic separation, where we have these magnetic beads that are tagged with antibodies specific for a certain bacteria, and we use those to pull them out of solution. Well, the Nugent group has been doing work at actually replacing those antibodies with bacteriophage and then using them to pull the target bacteria out of solution and then detect it. And some of the advantages for phage absorption is it's very efficient. Once it's done, it's irreversible, which is unlike antibodies. It's very robust. And its selectivity, compared to other methods, is high. But its cost to produce is pretty low. So there's a lot of advantages for using phage as a potential antigen to pull out bacteria. And so the scheme that they've developed there in the Nugent lab is kind of you take your sample, you add in your magnetic phage, and we'll watch the video play here. So you now have these phage that are tagged with magnetic nanoparticles. They'll bind to the surface of the cell. And then you go out and you apply your magnet to the side of your tube. And that will start pulling those cells tagged with phage out of the solution to the wall of the tube. And then you can aspirate what's left, and then what's left on the side of the tube is now concentrated cells. And then you can either pair this with a detection method where you have the phage also producing a reporter enzyme or something like that. And here you can see these are, this is under the TEM, and here's some E. coli cells, and these little particles here are phage that are tagged with magnetic nanobeads. And they've also done some work with just microbeads that are magnetic, and here you can see the microbeads under the SEM attached to the E. coli cells. And so there's a potential here then to also apply bacteriophage for separation. And so these are kind of all the things. And talking about how this compares to immunomagnetic separation, you can see here with the microbeads, phage-based microbeads showed improved capture efficiency over antibodies. I don't have the slide here, but when we looked at the nanobeads, they were at parity with one another. But here there's some potential for improvement, right, over traditional systems. And so the outlook, right, for using bacteriophage to control foodborne pathogens, I think there's a lot of opportunities, but there's a lot of research that needs to be done. If you're going to be using bacteriophage in a food application, you really need to do some validation studies in-house to understand how those phages actually work in your food system, because I don't think you're going to be able to just grab something that's been published in the literature and assume that applies to your situation, because you're going to have to look at how it reacts in the matrix, what are the temperatures that it undergoes, what are the pHs, the salt conditions, those all will impact the efficacy of your phage. But there's great potential for researchers to go out there and really understand the biodiversity of phage and what opportunities that will allow us then to improve the use of this tool, which is really just one tool, right? We talked about biocontrol really being a series of hurdles, and one of the things is phage is not going to be, again, another one of those silver bullets. It's something that needs to be used in conjunction with other control methods to help you make safe food. And so with that, any questions? I'd like to again thank Dr. Martin Wiedemann and Dr. Sam Nugent at Cornell for a lot of the research that I highlighted here. So thank you very much.
Well, thank you so much for your nice presentation. I have two questions. When you apply to fight bacteriophage into the food, like, do you remember any MOI number? MOI number?
On those studies, well, you can kind of back calculate the MOI number
Yes,
from those.
very big.
But and they were very, so in those cases, they were very high MOIs, right? So those initial studies that I talked about in the, in the listics, they're like, over 10,000. But some of the studies were lower. So it depends, right?
It depends.
And, you imagine in a food application, hopefully, right? You have low levels of pathogens, right?
Okay.
And so you still, to find that Loch Ness monster in that large lake, right? You need a lot of phage to get there, right? So you're going to have an incredibly high MOI.
Okay. And then I have another question is that when you apply to the bacteriophage into the magnetic bead, so do you using any linker or just directly absorption?
Yeah, so they've done both. So you can then use some chemical modifications to then like attach the amine group to a carboxyl group on the bead. They've also done work where we've modified the capsid so that it expresses it expresses a protein that gets biotinylated by the E. coli, and so then you have a biotin on top, and then you take magnetic beads that have streptavidin, and then you can attach the phage to the bead using that connection.
Which one provide best result?
Off the top of my head, I don't know the answer to that. I have to go back to Sam Nugent and ask him that.
Okay.
Yeah.
Thank you.
Dan Vassar with Target. Are you familiar with the use of bacteriophages in the environment applications? So we talked about food, but how about things like floors or floor drains or other maybe non-food contact surfaces where they may be applied?
Yeah, that's a great question. And off the top of my head, I don't have any studies, but you're going to have these challenges of making sure that you get the right level of phage, right? Because also, you're not just applying phage to the environment, you're also applying things like water and sanitizer, which are going to dilute your concentrations. And these are some of the challenges where we really need some more research to understand what's going on there and how effective that is. Because it has potential, but it needs to be studied. Any other questions? Then I'll stop holding everyone back from the little reception. And, thanks.